So, good morning, everybody. I'm glad that so many of you managed to do it on Saturday morning. My name is Joseph Zohar. I'm the president of CINP, and this is my great honor and pleasure to chair this session with the director of NIMH, Joshua Gordon. If I start and read all the publication and the awards that he got and all the achievements that he managed to achieve so far, it's going to take the entire hour. So instead, I was thinking about putting my personal experience. More than 30 years ago, I was starting my career in psychiatry, and then somebody suggested to me to apply for postdoc at NIMH. So I applied, got the Fogarty Award, and then I spent 3 years at the NIMH. And I can tell you, and especially the early career in this room, that this was extremely useful, pleasant, productive time for me. So if there are people here in the room that consider where they should do their postdoc, NIMH is certainly, based on my personal experience, very good place to start thinking of. Okay, so this is instead of formal introduction to Gordon, and I'm very pleased to ask him to come to stage and talk about the challenges and opportunities in mental health. Please, Joshua.
Thank you, and good morning, and thank you for joining me this morning for this plenary. It's really a pleasure to be here in Tokyo, one of my favorite cities in the world, to be able to talk to you about the National Institute of Mental Health and our view on challenges and opportunities in mental health research. Just so that we're all on the same page, although I'm sure most of you know, I'll give you a brief overview of the NIMH. And then I want to talk for a moment, especially given that this is the 75th anniversary of our institute in the United States, to talk about progress and successes of the research that we've supported. And I think it's important to do that, especially in our field where so many people, both within and outside the field, are quick to recognize our failures. It's important that we also recognize our successes for several reasons. First, we have to make sure to encourage the next generation to pursue research, because otherwise there will be no successes in the future. And second, if the public and policymakers and funders are going to support mental health research, they have to understand that there are successes at the end of it. Then after that, I'll talk about research priorities in 4 different areas: basic biology, computational approaches, systems of care, and mental health disparities in the US and elsewhere. I'll say I have nothing to disclose. That's one of the major advantages of working for the government, is you can't take anything from anyone else, even breakfast. All right. So the NIMH, as most of you again know, is the lead United States government agency for research on mental illnesses. We support over 3,500 research grants and contracts at institutions and universities around the US and indeed around the globe. And in the US, on our Bethesda campus, we have about 650 scientists performing research directly funded by NIMH in the intramural research program. Our vision is a world in which mental illnesses are prevented and cured, and our mission to achieve that vision is to transform the understanding and treatment of mental illnesses through basic and clinical research, paving the way for prevention, recovery, and cure. We articulate more of the details of this mission in a strategic plan for research, which is a living document on the web. I encourage you to visit it to learn more about the priorities that I'm going to talk about today and in other areas as well. And as I mentioned, we are in the midst of a year-long celebration of our 75th anniversary. We were founded by an act of Congress 75 years ago. This includes a range of different symposiums, lectures, and sessions at scientific meetings, as well as standalone events that we've been hosting in the Washington, D.C. area that are also broadcast on the web, that are available also as recordings after. I encourage you to visit our website to learn more about those symposia. The next one is going to take place on September 20th in Washington, D.C., but it's also available for simultaneous watching online. There's a series of podcasts and videos, etc. as well. I encourage you to visit our website to learn more. Now, in those 75 years, we've been supporting quite a lot of research. And as I mentioned, I think it's important to think and talk about the progress that we've made. One of the most famous members of the NIMH was the Nobel Prize-winning intramural researcher, Julie Axelrod, who, along with two other researchers, received the Nobel Prize for research on the chemistry of nerve transmission, and specifically on the mechanisms of reuptake of neurotransmitters by nerve cells. Now, on the one hand, this was a tremendous basic science discovery that all of us who've gotten PhDs or even studied neuroscience in undergraduate school know because of its importance for establishing how neurotransmission is regulated. But it also played key roles in understanding how the very first antidepressants worked, showing that they inhibited reuptake of various monoamines, and permitting then the development of more specific reuptake inhibitors like the selective serotonin reuptake inhibitors, which became even better antidepressants than the original ones. So this very, very basic science research carried out at the NIMH has to be connected to many of the treatments that we are using today. Because of those fundamental discoveries, we were able to improve on the original discoveries, which were made more or less by chance. These include, of course, advancing from MAOIs and TCAs to the various options that we have for our patients today. And while none of these are magic bullets that will cure every patient, we have to recognize that these gradual improvements over time have created opportunities for many, many patients to recover that wouldn't have otherwise been able to, or many patients to stay on their medications when they could not tolerate them. Now, there is a more recent example of a more direct translation of basic science into new medications that really came from a very theoretical approach that I think we can also lay claim to. And that's the drug brexanolone, and now zuranolone as well, for the first drugs approved by the US FDA specifically for postpartum depression. This is a graph of a paper by a number of scientists at the National Institutes of Health, including members of the NIMH Intramural Research Program, that demonstrated that the neurosteroid allopregnanolone could enhance GABA currents in neurons. This discovery, coupled with the knowledge that allopregnanolone is a derivative of progesterone, and then the demonstration that levels of allopregnanolone rose during pregnancy and then dramatically decreased at parturition, both in animals and in humans, led to the hypothesis that these rapidly decreasing levels of allopregnanolone might be a contributing factor to postpartum depression, and to the hypothesis that supplementation of allopregnanolone during postpartum depression might be a good treatment. Some 30 years later, these results were translated. I should say first, it was shown that this could work in animals, and then they were translated into human beings, with brexanolone being a preparation of allopregnanolone that could be used intravenously. And intravenous infusions of this drug showed that they could reverse severe hospitalization requiring postpartum depression in about half of the women it was tried in, and in a matter of days as opposed to the usual multiple weeks. This is the first true bench-to-bedside drug from my perspective in psychiatry, and it's an important recognition of the fact, number one, that basic science advances can and do help our patients, and number two, that the timeline for that is quite protracted. Now, given that, I think it's important that NIMH continue to support research in basic neuroscience and other basic biological studies of relevance to mental health. So I'm going to start, when I talk about our current priorities, with some discussion of that area. And then I'll move on into other priorities as well. For a long time, we have known that mental illnesses are, at least in part, due to our genetics. This has been established from many, many family studies that demonstrated that the genetic risk for illnesses like schizophrenia and bipolar disorder and autism are considerable. But over many decades as well, there were many failures in terms of identifying the risk factors, the specific genetic risk factors. That changed about 20 years ago when we began to recruit larger and larger numbers of individuals into genome-wide association studies. And these genome-wide association studies was the first approach that established many, many, many risk factors for illnesses like schizophrenia. This plot on this slide is a plot of all of the 280 some odd places in the human genome that are statistically significantly linked to your risk for schizophrenia. Each one of these green diamonds represents one such place in the genome. This remarkable breakthrough has been repeated for several other mental illnesses as well, and work is ongoing for others. There are several important points to consider as we think about this graph. First, we now have hundreds of clues as to why individuals develop schizophrenia. Second, each one of these clues is in itself another biological puzzle that we need to solve. And third, there are lots more out there to be found. Now, that deciphering the meaning of these biological clues represents a tremendous challenge for our time. There are hints, though, that this challenge can be met. For example, systems biology studies have demonstrated that the most likely genes that underlie the risk in these loci tend to implicate specific biological processes in specific cells at specific times. For example, the gene set implicated by these genetic locations is overrepresented with genes that encode for proteins that are present at the synapse, both postsynaptically and presynaptically, and expressed in neurons early in development. That demonstration, first of all, underscores the long-held hypothesis that schizophrenia, although it's an adult-onset illness, is an illness with origins early in neural development. Second, it tells us that if we can understand more about the roles of these risk factors in synaptic development in particular, we may be able to unravel some of the reasons why people develop schizophrenia. How can we do that? The brain is an organ which is inaccessible in adulthood, much less in childhood. Well, again, basic neuroscience studies have been enhancing our ability to answer these questions, not just genetically, but from a neural circuit perspective as well. Many different labs, funded some by the NIMH, others by the NIH's Brain Initiative, have developed tools and technologies to allow us to observe neurons and other cells of the brain developing along a fairly typical time course in a dish. We can study these cells in vitro to the point where they approximate developing brain tissue of about full gestation or maybe a little bit into the first year of life. These cells can be studied in a myriad of different ways, including developing organoids, or small balls of tissue that have multiple different neural cell types, which can form neural circuits and have functional neural activity. But it's been very challenging to get these organoids to develop into full-fledged adult-like brain tissue. One approach developed by an NIMH-funded investigator, Sergei Pasca, has taken these organoids and implanted them into mice to create mosaic organisms with normal mouse brain tissue, but also human brain tissue embedded as well. And this permits the organoid to survive later into development and develop more complex neural properties. A study done by the Pasca Lab has compared the structure of neurons developing these organoids growing up in mouse brains between, for example, individuals with no known large effect size mutations contributing to mental illness and individuals with Timothy's syndrome. And the Pasca group was able to show that the complex elaboration of dendrites, of neurons growing in these organoids, was diminished in Timothy's syndrome's patients. And with this diminished array of dendrites, there was a greater density of synapses along those dendrites. Now, it is challenging to understand the role of these cellular phenotypes in the cognitive and other behavioral phenotypes of Timothy syndrome individuals. But that's not the point of developing mouse models. We're not trying to mimic the full spectrum of Timothy syndrome. We're trying to understand the biological properties imbued by the mutation that results in Timothy syndrome, allowing us to test methods which might reverse these phenotypes. And indeed, that's exactly what the Pasca group has done now. They've developed an oligonucleotide approach to try to lower the expression of genes that are affected by the Timothy syndrome mutation, and they can show that they can rescue the complexity of these dendrites by these antisense oligonucleotides delivered to these organoids growing up inside of mouse brains. So these complex technologies that allow us to study development in human tissue through moderately advanced stages of the developmental process has the potential to reveal therapeutic avenues that could help patients in the not-too-distant future. We need to do this not just for Timothy syndrome, but for all of the many mutations which are relevant for mental illnesses. We need to understand the role of the genes implicated by these mutations in protein networks, in cellular structures, in synaptic and other functions of those cells, and then how those synaptic functions alter circuits and then eventually behavior. So NIMH has been, for over a decade, supporting research that will develop scalable and systematic neurobiological approaches to interrogate these genes. Phase 1 already concluded, has developed high-throughput assays such as the one that you just saw, involving cells and organoids, that allow us to probe the effects of these genetic mutations across multiple gene and protein networks and cellular functions. Phase 2, under development right now, are medium throughput assays that can take the most promising approaches developed in Phase 1 and study them in greater depth. And Phase 3, which we reserve for the future at this point, is developing lower throughput assays to study these processes in mice and primates and develop novel therapies based upon them. This approach is one that we feel will give us the best shot at deciphering the complexity that genetics has revealed in the origins of mental illnesses. Complexity is a theme, of course, throughout our understanding of mental illness. And that complexity exists not only at the genetic level, but at multiple levels of function and dysfunction in individuals with mental illness. we need better theoretical and computational approaches to unravel that complexity. So I want to talk to you next about our research priorities in computational psychiatry, which we have been advancing for the entirety of my term here so far at NIMH of 8 years. One of the most promising avenues of theoretical and mathematical approaches in psychiatry is the notion that one can use these approaches to make predictions even in the absence of understanding. For example, an NIMH-funded investigator, Dr. Amit Etkin, developed an algorithmic approach to sort electroencephalograms from individuals suffering from depression into different types, and use this algorithm to make a prediction about how much an individual would improve with their depression when given a particular antidepressant drug, sertraline, a serotonin reuptake inhibitor. And Amit and his group showed that this algorithm was reasonably successful at predicting the degree of improvement across 4 different studies of individuals given sertraline. Dr. Etkin then founded a company, which we also fund through our small business grant process, to exploit this EEG biomarker to develop novel treatments. But from my perspective, these kinds of predictive algorithms could be helpful at solving one of psychiatry's most vexing problems for currently active clinicians. What treatment do I offer my patient when they come in suffering with depression? We have many different treatments that we can offer. We have drugs of different chemical classes that target different molecules. We have psychotherapies of different kinds. We have brain stimulation methods, both invasive and non-invasive. And when study after study has been attempted, no treatment has proven better than any other treatment, with the possible exception of electroconvulsive therapy, which tends to help more people than any of the others. And even with all those therapies, many, many patients still do not achieve remission on these treatments. When a patient walks into my office and I diagnose depression, I usually have a discussion with them about which side effects they want to avoid. I have no other way of deciding which of these many options might work for them. Wouldn't it be great if we could do an EKG and not just predict the likelihood that they will respond to one treatment, but predict that they will respond to one treatment better than another? And in fact, although it wasn't done prospectively, Amit was able to show that the same algorithm that predicts response to sertraline predicts lack of response to a brain stimulation therapy, repeated transcranial magnetic stimulation, suggesting that if the patient had the signature of positive response to sertraline, that they would be less likely to respond to TMS and vice versa. So by doing a simple EEG before treatment, I might be able to recommend to my patient, you know what, sertraline's more likely to work for you than TMS. This kind of approach we need to test prospectively, and we need to demonstrate will result in faster responses and savings in terms of our patients' suffering and in terms of healthcare system dollars. And so the NIMH has developed an innovation funnel approach to biomarkers that will allow for treatment prediction for depression. We're funding a number of these at an early stage, accruing pilot data to suggest that they might work to distinguish the likelihood of a patient from responding to at least 2 different antidepressant therapies. Then the ones that are successful in that pilot phase will advance to a prospective small academic trial, and the ones that are successful in those will move on to a community-based phase 3 trial that would permit FDA approval of the biomarker for treatment selection. Of course, we're not just interested in biomarkers for depression, we're interested in dissecting the heterogeneity of all psychiatric illness. And so we've embarked on an even more ambitious project to try to categorize our patients not only by diagnosis, but also by any other information that we can reasonably obtain about them. This project, called the Individually Measured Phenotypes to Advance Computational Translation in Mental Health, or IMPACT-MH, encourages investigators to develop cohorts of individuals which will undergo some form of deep phenotyping. It could be imaging, it could be EEG, but most importantly, it includes behavioral phenotyping, because this can be done at scale for very cheap. We can then ask these investigators to compare and the ability to make clinical predictions about their patients based upon the clinical record alone, and compare that with clinical record plus the deep phenotyping, and quantify how much better can we do at predicting clinically relevant phenotypes based upon this additional information. Now, clinical relevance can be defined broadly. It can be about predicting what treatment might work best for a patient. It can also be about whether someone's going to have a longitudinal relapsing and remitting course, something more acute, or something more chronic. We hope that through these efforts like the Innovation Funnel and the IMPACT-MH program, we will advance psychiatry into a field where it's no longer guesswork as to what kind of a patient we are facing and what treatments might work better for them. But we have quantitative data to be able to have discussions with our patients about their prognosis and treatment. One resource we're taking advantage of in this effort is the NIH All of Us research program, an effort in the United States to accumulate 1 million people who donate their electronic health records as well as agree to be recontacted for further studies. We have begun rolling out behavioral phenotypic tests to them in a project we're calling Exploring the Mind. I've done this myself. It's really fun. If you're an American and signed up for the All of Us, I encourage you to do it. These are gamified behavioral tasks that people can do at their computer or on their smartphone that can be used and then and then linked to the electronic health record data that is being released from All of Us. Already, 60,000 people have taken these Exploring the Mind tasks, and the first release of data from the ETM task will take place in early 2025. So these are some examples of trying to decipher complexity in the mechanisms and effects of mental illness at the basic biological level and at the more phenotypic level using computation. We also need to decipher or unravel the complexity of delivering therapies to our patients, especially in the United States, but also true around the globe. The systems of care that we have for mental health are incomplete and not up to the task of delivering care to all who need them. And so NIMH sponsors research at the systems level as well. One of the most profoundly impactful pieces of research in the last couple of decades has been the Recovery After Initial Schizophrenia Episode, or RAISE study that was funded, for the record, by my predecessor, not by me. This study built on work done in Melbourne, the site where we'll be visiting next year, by Pat McGrory and others, and in other locations around the globe, showing that an intensive package of therapies delivered to individuals suffering from their first episode of psychosis could improve outcomes. The RAISE study took that package and applied it to community settings, real-world delivery settings in the United States, and showed that individuals with a short duration of psychosis could improve much more rapidly when they received this full package of care compared to usual care in those same clinics. Based upon those RAISE results, the U.S. Congress appropriated dollars to fund programs around the United States that use that package of care, termed coordinated specialty care in the United States. And there are now over 340 such clinics located around the United States, funded in part by these federal dollars and in part by state or local funds. These 340 clinics serve well over 3,000 patients a year. Over 30,000 of them have been have been treated through these first episode psychosis clinics since the advent of the program a mere 5 years ago. These programs are changing the face of care for individuals with psychosis in the United States. But we don't know how well they're working, and we want to be able to build upon these successes to improve them beyond it. So, 5 years ago, the NIMH took these increased first episode psychosis clinics and set about to develop a learning healthcare network within them that can teach these clinics how to improve care in real time. There are 8 different networks of clinics comprising well over 40 clinics, 40 of those 380 clinics, which are participating in the EpiNet network. All 8 networks submit their data to a national coordinating center. These data that they are submitting include patient outcome data, but also provider data specifically about fidelity to the coordinated specialty care. And we can ask, using this comprising data, how well are these clinics doing at improving the lives of patients? Which clinics are doing better and which clinics could use some improvement? And in particular, what elements of coordinated specialty care are most important to ensure fidelity to? These data are not just published in academic papers, although that is part of what these networks are doing, but they are also returned to the very clinics that are participating in the network, so those clinics can do continuous quality improvement. We'd love to expand beyond psychosis. The United States has embarked on a bold experiment answering the mental health crisis we're experiencing by funding more and more community behavioral health clinics around the United States, well over 500 as of 2022. And so we're launching collaborations between these community behavioral health clinics and academic centers to try to understand what kinds of treatments are being applied here, what kinds of research questions can be asked, and how we might turn these clinics into learning healthcare systems as well. Now, these systems of care are not only places where we can improve upon existing therapies, they're also places that we can harness for translation. However, it's hard even for a large governmental organization with a $2 billion budget a year to do this on its own. And so we partnered with other funding agencies from around the globe, as well as with pharmaceutical companies, to develop a public-private partnership interested at developing better treatments for psychosis, specifically early in the course of illness in the clinical high-risk phase. This project, called the Accelerating Medicine Partnership for Schizophrenia, is a project of the Foundation for the National Institutes of Health, which supports this public-private partnership on behalf of NIMH. Researchers have developed 2 different networks of care, one located in the United States and one with sites around the globe, to develop biomarkers for individuals in this clinical high-risk state, and what we're about to do, to embark on clinical trials of novel therapeutics that will help people in this state lead better lives now and reduce their risk for psychosis in the future. Finally, in the United States, we can't just talk about a unitary mental health crisis. We have to recognize that mental health care is different for individuals in different places and with different means. And of course, from a global perspective, this is also true. In the United States, we have access to all those different drugs that I showed you for depression. It's not necessarily true in lower and middle income countries. Moreover, we have access to, although it's an undersupply, many, many more psychiatrists per capita than many other countries around the globe. So we need research that addresses mental health disparities both within the United States and around the globe. Part of our strategic plan is specifically to address mental health disparities because we note that individuals of different races, different gender, sexual and gender minority status, geography, and socioeconomics have different outcomes for mental health, both in the United States and abroad. And our strategic plan specifically notes that it's important for us to engage with communities in collaboration for this research so that we can better understand their needs and develop systems of care that are appropriate for those communities. One of the ways that we're doing it is also recognizing that many of the circumstances which cause mental health disparities are the same circumstances that cause disparities in outcomes for other health care issues. If you are impoverished or living in a rural area in the United States, you don't only have worse outcomes from mental health, you have worse outcomes for diabetes, cardiovascular health, asthma, and a whole host of other health problems. And so when NIMH began to consider how can we begin to study not just the problem of health disparities, but develop solutions that will reduce these disparities, how can we do that? We realized that we can't do it on our own either, but we have lots of other institutes at NIH. There are 27 different institutes and centers at the U.S. National Institutes of Health, each of them concerned with a different organ system, clinical problem, or population. So we collaborated with those other institutes and centers to form the Community Partnerships to Advance Science for Society, or COMPASS program, which is supported in particular by the NIH Common Fund, a fund dedicated by Congress to help encourage these kinds of trans-institute projects. The COMPASS program funds in a really interesting way. So as I mentioned, we support 3,500 grants and contracts to institutions and universities around the globe, but virtually all of them go to academics or research institutes. COMPASS program does it differently. We fund the very community-based organizations that are involved in trying to remediate problems in their communities, social service organizations, and recreation organizations, agencies, nonprofits that are in communities trying to help. We asked these organizations to come up with health problems that they're trying to solve and interventions that they want to implement in their community. And importantly, these interventions, we ask them not to focus on the healthcare system or novel healthcare interventions, but rather structural interventions in their communities that address many of the causes of disparity. We know, of course, that socioeconomic status is one of the causes of disparities for health, but it's not the lack of money per se, right? It's the fact that people with less money can buy less healthy food. They don't have greengrocers in their neighborhoods. They don't have parks in their neighborhoods. They face greater pollution, fewer trees to filter that pollution out. Lots of different contributing factors that have nothing to do with the healthcare system. And so we asked these community-based organizations to develop interventions that targeted these structural factors in their communities. We paired these organizations with hubs that were indeed awarded to academics and other research institutions that provided the necessary expertise for experimental design. We funded 20 such projects, each one, every 5 of them paired with a hub and a data coordinating center as well. These awards went out last year, and we look forward to the results. But one of the other interesting things is that while we asked the organizations to target specific health problems, we tasked them as well with working with the Data Coordination Center to ensure that their experiments measure not just their health outcome of interest, but health outcomes across a broad range of areas. So one community organization that wants to target asthma will also be looking at diabetes, will also be looking at depression, suicidality, and other mental health outcomes. This will allow us to compare and contrast across these 20 different projects as to which ones affect which areas of health and which ones give us the biggest impact. We're really proud about the COMPASS program. We're really proud about all the research that I talked about today, and I'm happy to answer questions and have a discussion with you with the remaining time. Thank you.
Thank you so much for this excellent overview. Yes, please.
Yeah. Thank you, Dr. Gordon, for a very great presentation. And I'm Jun Miyata from Aichi Medical University. And my question is the relationship between current project of NIMH and the RDoc, formerly I think it's the basis of research projects of NIMH. So I'd like to know the difference, the relationship.
Thank you. So the RDoc program, Research Domain Criteria, is a project of the National Institute of Mental Health. It was conceived of by my predecessor, and we're continuing it as well. So the basic idea behind RDoc is that by understanding the behavioral and cognitive processes that the brain is trying to perform, we will do better at, number one, understanding our patients, and number two, dissecting the heterogeneity of mental illness, and number three, delving into the neurobiology thereof. There have been several successes that we need to tout of RDoc. Perhaps the biggest one is the recent demonstration that you can measure the brain relationships of anhedonia, right, of the lack of pleasure that one sees typically in depression, but in many other mental illnesses as well. And that you can target anhedonia with a kappa opioid antagonist. And there are now, after that demonstration, 2 different companies developing kappa opioid antagonists currently in phase 3 trials, I believe both for depression. There are other successes as well that I can tout. The essence of RDoc is deeply embedded in the precision psychiatry project that I mentioned, Impact MH. What I would say that we have done in the last 8 years is try to transform RDoc from a descriptive, categorically based approach to dissecting behavior into one that is more quantitative. We have asked investigators to test, for example, function in different RDoc domains and try to examine whether functional measurements within domains are more related than functional measurements across domains, for example. And Impact MH seeks to take functional measurements within RDoc domains and ask precisely how much more information do we get about our patients from adding things like RDoc concepts to our description of patients compared to using diagnoses alone. As my predecessor said many times, and as I will reiterate, RDoc is not meant to be a diagnostic system. It is not meant to replace a diagnostic system. It is meant for research, and what we're doing with Impact MH is also trying to understand, adding it to clinical diagnoses, does that improve our ability to make clinical predictions?
Yeah, thank you very much.
Yes, Tony.
Hi, Josh.
Hi,
Great
Tony.
talk and very encouraging information. I wanted to express a concern. 30 or 40 years ago, when people talked about disorders like schizophrenia, it was always gene-environmental interaction, that it wasn't genetically determined, but genetically associated. Because we know that at least a third of schizophrenia is non-familial, that it can be associated with childhood trauma even outside of a family history, and even identical twins only show a 50% concordance. Now, Eve Johnstone did some nice epidemiological studies looking at children at risk for schizophrenia and found the ones that showed hyperresponsivity to stress tended to be the ones that converted. And in our animal models, what we find is whenever we do a developmental disruption, it's not the developmental disruption that causes the adult pathology, but instead it makes them more susceptible to stress during critical periods of development. My concern is that by focusing exclusively on genetics, especially in isolated systems, we might be missing an important variable in the pathophysiology.
I think your concern is appropriate. I wouldn't say might, we are, right, missing. If we're looking at organoids, although certainly there are people trying to look at stress in organoids, I would argue it's very different to stress an organoid with a chemical or than it is to stress an organism. In particular, stress, as you know, occurs in that early fetal development, or I should say affects fetal brain development. The talk today focused on our efforts in the genomic space. And part of the reason why I wanted to focus on that is because it's an area where we've had tremendous success in understanding, and there's a lot of potential. But it doesn't cover all of what we're doing in that space. And yes, indeed, focusing on circuit models that are based on genetics will not give us the full picture. I would add one thing in that regard, though, Tony, which I'm sure you know, and that is that it's been very challenging to move beyond stress into specific mechanisms. And where we've done so, we haven't been able to test those hypotheses in humans as well as we would like. So for example, a lot of work on the HPA axis and all the various hormones and glucocorticoids, et cetera, and that hasn't yet developed into a tested hypothesis in humans showing that that can actually affect, say, the outcomes with schizophrenia or, for that matter, with depression. Genetics gives us a potential way in to get at the biological pathways involved in stress if we can combine these models. And I know you know this because I know you do that, right? But as we get more sophistication and understanding of the circuits from the genetic and circuit combination, adding in stress will be important to try to get at the mechanisms, not just stress, but other environmental factors as well, to get at the mechanisms by which those affect disorders like schizophrenia. So thank you for bringing that point up.
Thanks.
Yes, please.
Thank you, Josh, for an excellent summary of NIH-funded research. I'm Tadafumi Kato from Juntendo University. And in addition to the studies you introduced in your presentation, many studies using animal models, especially mouse models of mental disorders, have been published in the last several decades. But unfortunately, none of them led to the innovation of psychiatric practice yet. So do you think that this situation will continue another decade, or those basic neuroscience studies will finally translate it into clinical practice?
Well, thank you for especially for including that word "yet." Right? I would argue actually that, I mean, it was kind of lucky, but the brexanolone actually grew out of animal data. So let's be clear. I think there have been animal research results that have successfully translated into human therapies. But for every brexanolone, well, really, that's the only one, there's many, many failures, right? First and foremost, time is really important. Those studies on animals were done on rats in the late 80s and early 1990s, and the therapy was approved in 2019. So it takes some time. But I think there are other things that we can do to try to improve upon translation. First and foremost, we have to make sure that we're translating at the right level, right? So, of course, famously, you know, Fragile X was thought that we might be able to develop therapies based upon long-term potentiation circuits from a mouse, and that didn't work. Is that the right level to be translating? Should we be focusing more at the cellular and molecular level when we're trying to translate? I don't know, but we need to figure out what the right level to translate is. The second thing that I think we need to do as well is to develop more non-human primate models as intermediaries. I mentioned Timothy's syndrome, where we're studying these cellular effects in human tissue embedded in mice. There are also things like Angelman syndrome, where people have been studying genetic ways to reverse the genetic deficit in mice, and showing that at particular times of the development of the mouse, you can fully restore the behavioral and circuit effects of the Angelman's mutation. But it's really hard to translate that into a place and time where you would do it in humans, although there are companies already that are trying to do this. I would argue that it would be better to develop a non-human primate model and figure out when in the development of that non-human primate can you intervene. And especially if that agrees with the mouse, that promises better translation in humans. And of course, if it disagrees with the mouse, then you might need to then move into human tissue to make sure that the non-human primate model is more accurate. So I think we need a more staged process to translate the information that we learn from mouse into human. Some things may translate directly, but I'd imagine that most of what we're trying to do needs to be translated through a higher animal model or through human tissue itself.
Thank you.
Yes, please.
A question here to follow one of the previous question. What is the current mental health position in terms of dissecting multifactorial mechanism of mental health disease? Because it seems like in a genetic study you can identify very impressive p-value, but effect size is very small, even if you consider polygenic scores. However, more evidence to support genetics, environment interaction, and environment is really complicated for humans. Not only stress, but also what we eat, where we live, radiation, temperature. A lot of things we can manage more manageable than genetic risks. So if we can identify the overall mechanism for mental health, I think it's much easier for us to consider system healthcare instead of in fact, you have realized mental health disparity, and that also reflects the multifactor ideology situation. So can you tell us about your position for this kind of strategy?
So first, let me react to that last bit. I told you how we're studying interventions that can remediate environmental factors involved in mental health risk through the COMPASS program and other programs that I didn't get a chance to tell you about. So it's important to recognize that while we are focusing on genetics, we're not doing that to the exclusion of other factors. We invest heavily in stress. We invest heavily in trying to understand other environmental factors and their impact on biological systems, and we invest heavily in research that seeks to counter risk factors at a systems level. So I would say you cannot focus on any one pathway towards novel treatments. Our patients need us to do better, and we need to attack that from all possible paths. Second thing is I want to react to one of the small facts that you mentioned in the beginning, which is an important fact. Many of these genetic risk factors are a very small size. So first, yes, that's true, but we also have 10 known large effect size mutations in schizophrenia that involve single genes that we can study from a biological pathway, where the challenge of understanding them is simpler, although still not simple. In autism, the story is even richer, with probably 200 different large effect size mutations of single genes that we need to study. So I think there are simpler paths than the small effect size mutations that you mentioned. The other thing, as I noted in my talk, is there are systems biology approaches that may give us the ability to translate even the small risk factors into actionable biology. I'll add one other thing that genetics does, and I hinted at this in my response to Tony Grace's question. And it gets to the heart of what you were asking. How do we study these non-genetic risk factors with tools that will give us a greater ability to understand the impact and to develop interventions based upon it? So on the one hand, deeper phenotyping of our patients should not be limited to behavior. It should also be about exposure. And there are a number of different attempts to look at, in more complex and deep ways, at the exposures our patients have had that go beyond the studies that we already know about, like adverse childhood experience scales, and, and other things. One can do it very simply, looking at zip code, for example, which has many, many correlates. Your postal code has many, many correlates to environmental variables. Or one can do it more complexly by, say, harvesting baby teeth and looking at the deposition of heavy metals at different ages of the tooth development, which gives you an insight into exposures that individuals had in their infancy. So there are lots of different approaches that are being pursued by NIMH and others in that regard. But genetics gives us another opportunity where we can ask, how do these exposures affect the risk engendered by genes? And that gives us a chance to look at gene-environment interaction, but most importantly, it gives us a biological pathway into understanding these environmental risk factors in a novel way. So if we know, for example, that your risk engendered on mental illness by heavy metal exposure early in life works through, I'm going to make something up, a phosphatase inhibitor. That gives us a biological clue and a pathway to treatment that we didn't otherwise have. And you can do that by combining deep phenotyping and genetics, and it's something we're really trying to encourage.
Thank you. Last question. Imagine that we got trained to the future, and our next stop is 2030. What would happen then?
2030, not much. That's too soon.
So let's move to 40.
I think we're going to have, maybe not by 2040, but the objective behind the computational predictions, which are really probably the most near-term that we can have, right? The objective behind our computational prediction is that we'll be able to accumulate, sorry, let me start over. We're going to make our clinical diagnoses like we usually do. And then we're going to be able to say, well, you have depression, but in order for me to know how to treat you, I want you to take these 6 behavioral tests and do an EEG. And then I'm going to input that information into my phone, and my little doctor's helper will spit out the probability that that individual patient will respond to SSRI versus cognitive behavioral therapy. And I'll be able to have a conversation with them. SSRI is more likely to work for you, but it has these side effects. CBT is not as likely to work, but it may work for you, and it has this cost and this problem of time that you have to invest in it. Let's have a conversation about which you want to try first. So that's what I hope we'll have in maybe 2040. That might be a little ambitious.
Thank you very much.
Thank you.